Hello, everyone. Welcome to SDIS monthly lectures. So to all the members of SDIS who are here, as well as the guests and members of affiliate organizations. You're all welcome. And we have an exciting talk for you today. But before introducing our speaker, I'd like to update you on coming events. On March 20, we'll have Dr. Monty Marshall, who is a retired professor of geosciences at SDSU. And he will speak to us on some marvelous discoveries in modern astronomy. And on April 17th, our speaker will be Dr. Edward Watts, PhD, who's a professor and chair of the history department at UCSD, and will speak to us about interesting analogies between the Roman Republic and the United States of America. And it's a little scary, actually. There are some events that he will talk about which almost mirror exactly what happened in this last election in the United States, including the insurrection at the Capitol. So it's kind of scary. And as he points out, the Roman Republic lasted for 500 years before becoming a dictatorship. We have only 250 years, but I'm not sure we're as good as they were. So it's not clear what's going to happen. But in any case, that should be a pretty fascinating talk as well. So now our speaker today is Dr. Jerold Chun MD and PhD. He is a professor and senior vice president, neuroscience drug discovery at Sanford Burnham Prebys Medical Discovery Institute. He has made important contributions to our understanding of the brain and its diseases, including the discovery of genomic mosaicism in the brain and its involvement in the most common forms of Alzheimer's disease, with relevance to other brain diseases. In separate work, he identified the first lysophospholipid receptor, which is part of a growing class of lipid receptors underlying new neuroscience medicines, including fingolimod, siponimod, and ozanimod for multiple sclerosis, and an understanding of other diseases, including hydrocephalus, schizophrenia, neuropathic pain, and fibrosis. Dr. Chun receives his MD and PhD neuroscience degrees through the Medical Scientist Training Program at Stanford University School of Medicine. He then moved east to Helen Hayes Whitney postdoctoral as a Helen Hayes Whitney postdoctoral fellow at the Whitehead Institute for Biomedical Research at MIT. He then joined the faculty at UCSD School of Medicine, where he became professor of pharmacology and neurosciences and directed the neurosciences graduate program. He subsequently became department head of molecular neuroscience at Merck Research Laboratories, and then returned to academia as professor of the Scripps Research at the Scripps Research Institute, an adjunct professor at UCSD, before taking his current position. Dr. Chun has authored more than 300 scientific publications and has been recognized in Thomson Reuters' World's Most Influential Scientific Minds citation list. He is a member of numerous advisory, review, and editorial boards in both academia and industry. Today's topic is, does internal DNA editing cause Alzheimer's disease? Dr. Chun.
Oh, thank you, Alvin. That's way too much to have to even talk about as an introduction. And I'm pleased to be here, and I've been already very much impressed with the camaraderie of your group. And I'd like to share with you some scientific data on Alzheimer's disease that I know many of you actually have the background to understand this type of work, but I'll be taking you through some of the basics for those of you that need a little refresher, and that it may represent a new way of looking at Alzheimer's disease. So let me see if I can get this to work here with the screen share, and we'll commence. And so the title is, Does DNA Editing Cause Alzheimer's Disease? And I'm located over at Sandford Burnham Prebys. And for those of you that haven't been by, we're across from the Torrey Pines Golf Course. It's a small institute, private, nonprofit, but it does have quite a bit of activity scientifically. And I, of course, invite all of you to come and visit as this COVID craziness passes. And hopefully all of you have been able to get your vaccines or will soon get your vaccines. And then we can get back to some normality in how we interact. The work I'll tell you about today was done by a whole group of people in my laboratory, and they involved students such as, can you see my arrow here? I'll try to use an arrow. Okay, yeah. Perfect. Students, Christine Liu, Chris Park, Gwen Kaiser, and Diane Bushman, and then postdoctoral fellows, Ben Sidaway, William Romanow, Igor Segoda, as well as Rich Rivera, and then our informaticians, Tao Long and Yunjiao Zhu. We've had technical help from Grace Kennedy, and then the person that really drove a lot of this work was Mingshang Li, who's now a professor in Taiwan. So here we have the human brain, hundreds of billions of neurons and even more glia. It's estimated that there's perhaps 1 trillion cells within our single human brain. It learns, it remembers for a lifetime, it makes us human, and many diseases afflict it. The enormous complexity in organization has been thought to arise from and utilize a constant and immutable genome, but what might DNA editing to create genomic diversity provide? And I'll take you through a little analogy, a couple of slides that draw on some of what might be your artistic talents. So those of you that paint know that if you have a palette, you need multiple colors. However, if that palette had only one color, it'd be difficult to paint. And so that is a way of thinking about what genomic mosaicism could provide. Without it, you're stuck with one color in the palette, and then everything that comes off of that palette has to somehow be modified to produce the organization in a given picture. So it's analogous in the brain, we believe, whereby if you, instead of having this single color, had a range of different colors to choose from, i.e. different genomic states within each of the cells of your brain, then you'd be able to produce far more interesting levels of organization, of complexity, ways of storing information, and that process may have some what we would call unanticipated consequences. So that is the general rationale for looking at genomic diversity within the brain, is that it would give you a way to organize it that would be based on the most stable biological substrate in our bodies, DNA. And you may ask, well, why hasn't this concept really been explored more? It's not really something you see in the textbooks, or perhaps it's just starting to emerge there. And let's draw upon the art analogy once again. Here's a cup of brown paint. And once again, for those of you that paint, you know that that could be caused by every single droplet of pigment there being identical. It's brown. However, there's another possibility, which is you've mixed all these various colors together, and they too form that brown color. And what it turns out is that in the brain itself, it's this situation where you have myriad different colored droplets to form that, that brown paint, myriad cells with different genomic information to produce our brains. And those brains are thus mosaics of cells with different colors, if you will, of different genomes. And we refer to this as genomic mosaicism, where the DNA sequence varies in each cell, and it can indeed vary in each brain region. For those of you that know about epigenetics, that is where you have, for example, methylation of DNA. That does not change the sequence. So what we're talking about here is something that actually changes the very DNA sequence itself. You could say it actually changes the blueprint of our genes within individual cells of the brain. So that brings us to Alzheimer's disease. And here are a few facts. It's the most common cause of dementia, some $300 billion per year. in challenges to society, affects about 6 million US citizens, or about 2% of the population. We still do not understand the most common form, which is referred to as sporadic Alzheimer's disease, and it constitutes about 99% of cases. And this is despite hundreds of what are referred to, and many of you know about, so-called Genome-Wide Association Study genes, or GWAS genes. And so you'll read in the papers, oh, another gene has been found from doing various genetic studies. These are GWAS association genes. And I'll have more to say about that as we look at the next couple of slides. There are zero disease-modifying therapies that exist, despite many failed clinical trials that actually number in the hundreds and billions of dollars spent on research and development. And yet, despite all of that effort, we don't have a single drug or therapeutic that can actually modify the progression of Alzheimer's disease. However, there are some very striking links that have been identified. And the most prominent in my mind is the familial Alzheimer's disease link. And so I just mentioned sporadic Alzheimer's to you up here. We don't know the cause of that, but in these about 1% or so of cases, families that have certain genetic mutations will get Alzheimer's disease. And these are referred to as familial Alzheimer's disease patients. There are 3 genes that have been proven to be causal for familial Alzheimer's disease. 2 of them are presenilins that I will not have more to say about. But a third is amyloid precursor protein, or APP. And if you have mutations in that APP gene, or if you have more copies beyond the normal 2 that all of us have in our genes, that can be causal for familial Alzheimer's disease. So I'm going to spend quite a bit of time talking about, smiley face to tell you it's interesting, about APP and changes in this gene. So here's a summary, nicely placed to compare the causal genes, presenilins and APP up here. Once again, if you have these in your family with the appropriate mutations or copy numbers, you will get 100% Alzheimer's disease. But then we have all of these GWAS identified genes that have appeared, and some of these are familiar to you, such as ApoE4, in this case being a homozygote, having 2 copies of ApoE4, as well as those that have a single copy have some increased risk. But none of these different genes here are causal for Alzheimer's disease. So just because you have them does not mean you're going to get them. The risk is variable, and it affects as you can see, the percentage down here, a subpopulation. And as you can also see that, let's say 3% frequency in the population, well, that's clearly not going to be able to account for the Alzheimer's disease cases that we see that affect 2% of the population, and even less so for these common types of phenomena of genes that are in 20, 30, 40, half of people have these particular forms. So these are associations or risk factors that have been identified, but they are not causal. That's a very important point, because it means that the real mechanisms that we need to understand are not the risk factors per se, but how they might drive, for example, these causal genes. And it's, for example, one thought is that maybe some of these risk factor genes affect directly some of these causal genes, and there's some evidence for that in the literature. Let's take a closer look at the genome-wide association studies, just because they're so common, and you'll hear people talking about them all the time, including in the commercial sector with these types of Ancestry or 23andMe kits that are available. Maybe some of you have taken these. So the current way of looking for human disease genes are through these genome-wide association studies, and you take some peripheral samples, such as your buccal mucosa, taking some cheek cells, or taking some blood cells, and you isolate and analyze the DNA by sequencing. And the DNA sequences amongst these cells are assumed to be identical. Now, importantly, especially considering what I just told you about the brain having different genomes within each of these cells, the GWAS doesn't actually assess the DNA state from cells of the brain. They're coming from other parts of the body. And you can think about that as an analogy to our solar system, or to our galaxy. So here we have the Milky Way. It's got about 300 billion stars, and you can compare that to the numbers of cells in our body, which is about 37 trillion, or in our brain, which is about 1 trillion. And so if our brain is, say, Earth, it's out here somewhere in this the Milky Way, but you sampled something from way out here, and that'd be, say, 100,000 light years away, well, number one, you wouldn't be sampling a human, most probably. It'd be some alien, perhaps. But in addition, it's not going to tell you anything about really what's going on here on Earth, and certainly not yourself. And it's important to note that our bodies have 125 times more cells than all of the stars within our galaxy. So it's a big number and a lot of diversity that has not really been assessed. And here's a little cartoon. Some of you have seen this before. And why are brain studies, why do they rely on non-brain samples? Why do GWAS? And it's because you can do it. You just take a little sample and you run it. And so here we have this fellow. I'm looking for my quarter I dropped. Did you drop it here? No, I dropped it 2 blocks down the street. And why are you looking for it here? Because the light is better. And that's a lot of the philosophy that's driven these studies, is because you can do it, but it doesn't necessarily mean that it's going to actually take you to where you need to get. So let's take a step back here and look at a little bit of basic biology. And here we'll start with the chromosomes. We've got 22 plus X and Y to give you 2 copies of each. And 1 copy comes from your dad, 1 copy comes from your mom. And normally we thus have 2 copies of every gene, and your chromosomes, generally speaking, arise from your mom and your dad. Now in Alzheimer's disease, as I just mentioned, you can have increased copies that produce disease. And how do you get that? Well, you can get it through some duplications of genes like APP, or you can also get it by increasing another copy through a chromosome, as occurs in trisomy 21 or Down syndrome. So in Down syndrome, it's interesting. Everyone that has Down syndrome will have a brain that looks like an Alzheimer's disease brain by the time they're in their 40s. And there's some debate about whether it's truly Alzheimer's disease, but the general consensus is that it is Alzheimer's disease, or certainly Alzheimer's disease-like, and it's coming from the 3 copies of that chromosome 21. However, these explanations are not supposed to be relevant to that sporadic form, the common, the 99% that I mentioned, because when people have looked over the decades, they haven't really found evidence for either trisomies of chromosome 21 or for increased copies in looking at samples from individuals. A little more background, just reiterating that we have 1 copy from mom, 1 from dad to produce you, and those are fundamentally DNA. DNA comes from the discoveries, or were originally identified by Jim Watson and Francis Crick. Francis Crick used to be here in San Diego. And we start with that DNA molecule that then undergoes a process called transcription to produce an RNA molecule. And then that produces a protein through a process called translation. So it goes DNA, RNA, protein. And these are nucleic acid species, and these are made of amino acids. Very basic, but I'll have more to say about that in just a bit. Now I'm going to add to that information by telling you about some fundamental organization of that DNA. So here are those, a copy of a chromosome that we just talked about. And if you were to pull out the protein coding portion of the DNA, that is the gene, it has an organization that consists of information that, or sequence information that encodes the protein portion with intervening sections that do not. So these are called exons, these are called introns. And this is how a gene sits within the DNA. It sits over, in some cases, very vast stretches where 90% or more of the sequence will actually not be coding. It'll be this so-called intronic sequence. Then you have the smaller portion that produces the coding. and that coding must be turned into an RNA molecule and brought together to form the actual sequence that will give rise to that protein I showed you in the previous slide. And that process is produced here during transcription, which we just talked about, that forms an RNA molecule. Well, I simplified there because the first RNA molecule is a copy of this DNA that is present in the genomic portion of your DNA. It's a copy that contains those introns and the exons. And then a phenomenon known as splicing of the RNA occurs. And then you have a piece that forms the actual mRNA. And so this is what is encoding the proteins in our bodies. If you were to copy this mRNA itself and look at it and sequence it, you would form what's called a complementary DNA or a cDNA. And so much of what I'm going to show you in the rest of the talk comes from looking at some of these cDNA aspects that came from this type of an organization. And then ultimately, you would get translation to produce proteins. That was also shown in the previous slide. I'd like you to keep in mind this term, cDNA, complementary DNA. This is what all of us in molecular biology use for cloning purposes. And again, it's a copy of an mRNA molecule. We'll take one more little summary slide here. We start off with the cells of our body. Again, there's 1 trillion of these cells in our brain, 37 trillion in the body. Each one has a nucleus, and within that nucleus is where our genome is located. And from that nucleus would arise the chromosomes, which are basically packagings of the DNA. And if you were to unwind the packaging, you'd have these other organizations. I won't belabor them, other than to note that genes are encoded in them, and they would include the introns and exons that we just talked about. And these genes can be identified based on their nucleotide sequence. And we can use a number of different types of sequencing strategies to get the identity of that particular gene. However, there are other ways we can look at these genes. And I'm going to show you one example of that. And that is, you can actually paint regions of the gene using different types of probes that can identify these sequences. And so I'm going to show you a picture of an APP gene that has been painted in a single cell and actually a single nucleus, and done so in a way that you would only see it if you had more than 2 copies of APP. Why is that important? Well, I just told you that you don't get Alzheimer's disease in families unless you have multiple copies. In the case of Down syndrome, 3 copies, or in some of the familial cases, 3 or more copies of APP. So this could be a way that you could go looking around for cells that had more APP. And so here we have one such cell. The little green dot is only seen with more than 2 copies, and it's probably 3, 4, maybe 5 copies. And in addition, here's our positive control in red. These are so-called telomere sequences. The main point is that that controls for our technique. And we have evidence then that there is more than 1 copy of APP in the cell. So where did the cell come from? This cell came from a sporadic Alzheimer's disease brain. And I had told you earlier, well, we don't have more copies of APP in sporadic Alzheimer's disease. And in the germline, that is in all the DNA, the common DNA of all the cells, that may be true. But in cells of the brain, it appears that indeed we have more copies of that APP gene, suggesting that maybe this is a way that disease could be produced, just like it's produced in families with more copies. Okay, let's take one more step back to look at a very basic technique. This is called polymerase chain reaction, or PCR. I know all of you have heard of this. If you watch some of these detective shows, my daughter loves to watch things like NCIS and things that use forensics. They use PCR to identify a variety of rare species, and then that enables you to sequence them. And the way this works is you have a sequence of DNA, You stick on a couple of primers to it, and then a polymerase that tacks on more nucleotides enables you to magnify or copy the DNA. And it goes geometrically to copies, 4 copies, 8 copies, and you can go many, many, many copies to produce millions and millions of the same sequence that enables you to identify very rare very rare phenomena in the sequencing of your nucleotides. So PCR is a fundamental technique that we use, and I'm going to be talking a bit more about that next. So imagine that that little green dot that I showed you. How are we going to get at what's in that little green dot? And so a number of years ago, we designed a way to do that. And the first step that we took was to look at the RNA that was coming off of the DNA. And I told you that that's the way that basic Watson and Crick process going from DNA to RNA to protein occurs. Why look at RNA first? It's because you have more information or more copies of RNA coming off of the DNA. It's not one-to-one. It's actually one gene can produce thousands and thousands of RNA copies. So you basically are magnifying the signal coming off of the DNA. Then we can actually look at what the sequence of that is. And so we did just that. We took a cell or a few cells, and then we looked at its RNA sequence by producing a cDNA to it, that complementary DNA. And then we stratified it in what's called a gel. That's not so important. What is important is each of these bands here has a different size. And when we looked at the sizes of the bands that came from that RNA in these, in this case, neurons, it was surprising. And the reason it was surprising is that according to the literature, there should only be high molecular weight bands. There shouldn't be all these little bands. So we were surprised and quite intrigued. And the bands that we were expecting to see were actually these guys down here. So here you're looking at the splice products. So I told you here's the about the genomic DNA of the gene. Here's the gene for APP. Here are the introns. Here are the exons in these colored boxes. And when it's spliced, you have this single form of RNA that's produced. And it turns out there's actually other variants that aren't important for today's talk, but they are there for completeness. And so this is what we expected to see, something that's about 2,000 base pairs. But we saw these smaller ones, and so we cloned them out to identify them. And what we found is a whole bunch of little guys that were not previously reported. And they had some characteristics that were quite interesting. They were joined between their exons. So it was like you broke open the boxes that were present here, these exons, and rather than having them abut each other, you actually cracked them open and stuck them together again. These are what we referred to as intra-exonic junctions, or IEJs. And all of these small little guys had not been seen previously, and many of them, or the vast majority, I would say, are joined by these, we call them homology regions. Point is that these are novel sequences that had not been previously reported, and they had characteristic features. Moreover, we could see these features when we went looking for them in other types of databases. For example, we got a commercial library that was created in a very different way and could still see these types of species with the IEJs. So that led to the question of where are these coming from? And to make a long story short, we hypothesized that these could in fact be coming from the DNA. And so one would start off with the normal appearance of that gene, the one where we have 2 copies of that gene. But then something would happen within the RNA perhaps, or maybe in the DNA itself to produce these intra-exonic junctions. And then these could go back into the DNA and then produce their own RNAs. So let me review that again. You had the normal, we call it the wild type sequence up here. It then produces RNAs that have these new forms, and then it can be copied by a process that sticks it back into the DNA, and then that can again form new species. So we began looking at this possibility and to clone sequences out of the genomic DNA that might be a reflection of these new species that we refer to as genomic cDNAs. So remember, I told you to hold that thought in your mind about cDNA, complementary DNAs. So here we have them. They're here, except they're present in the genomic DNA as well. So we call them genomic cDNAs or gen cDNAs. So this is something new. It hadn't been seen or reported before, and it argued that there was something that was being created anew and stuck into our blueprint, stuck into the DNA. And we looked at it using a very special form of sequencing. I'll just mention it for those aficionados in the group. So the way to do these experiments is we would take a few cells, take their nuclei, and then PCR for these species, these genomic cDNA species. And we could do that because this particular PCR technique cannot interrogate the big size of your normal copy, but it could the smaller copies. We can then sequence them in their entirety using a process that's referred to as long-read sequencing, and it has other names like circular consensus sequencing or smart sequencing. That's not so important, other than to say now we had a way to actually go looking at many of these different potential species in the genome. So I'm going to pass through that. And here is what we found. And I'll focus your attention on this non-diseased circle plot, as we call it. So on the outside of the circle plot are the exons of APP, and it goes 1, 2, 3, 4, 5, 6, blah, 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 all the way to 18. That's the number of exons that are present within APP. And each of these little black dots that are seen in the different concentric circles are insertions, deletions, insertions, and single nucleotide variations. Basically, they're changes in the DNA sequence from what you would expect based upon the human genome sequences of humans. In addition, we could look at regions that contain the so-called A-beta region, which is important in Alzheimer's disease, because that is what's thought to produce these peptides that are necessary for the production of Alzheimer's disease, as the theory goes. And what you can see is that, yes, we do have little dots, not a whole bunch of them, but we do see them. In addition, I'm just going to point out in the very center, with the IEJs are there as well, the intraexonic junctions that are present. So now let's turn our attention to Alzheimer's disease. And once again, here are the 18 exons, and then you can see there are a lot more black dots. So there's a lot more going on in Alzheimer's disease. And these are logarithmic scales here, so, 1, 10, 100, et cetera. And so in some cases, we have hundreds or thousands more of these little black dots of these species that are present within the Alzheimer's disease brain. And in fact, there's about 10 times more diversity within this population as compared to this one. Moreover, you can see within these A-beta regions that there are black dots and that some of them have little red circles around them. So what are those little red circles? Well, remarkably, these red circles are actually identical to mutations that have been reported in families of Alzheimer's disease. And so we had talked about some of the mutations that can produce Alzheimer's disease in families. Well, here they are, except they're present on these genomic cDNA species, and they are present in sporadic Alzheimer's disease, the 99%, the most common forms. So this allows us, I believe, to tie in observations from families to the common sporadic forms, but it's not doing it on the normal genes that are present, the 2 copies, the one from mom and dad. It's doing it on these new genomic cDNA copies that are found within the brain cell DNA. So let's return to this picture where we have the central dogma. again, DNA, RNA, protein, and the 2 copies of each gene, one from mom, one from dad, into you. And what we can say is that, in fact, it appears we can have more gene copies and their variants via these genomic cDNAs. So rather than just having 2 copies, we can actually get more of them, and they can change through these genomic cDNAs or gen cDNAs. Moreover, the central dogma actually was modified very early on in its history by the work from David Baltimore and Howard Tiemann that discovered reverse transcription. So what is reverse transcription? It produces changes in RNA that go back into the DNA, and thus allows you to incorporate that sequence information from RNA into the DNA. And as you recall from the previous slide, it was thought that it always goes DNA, RNA, protein. In fact, it appears that RNA, or it's not appears, it's known that RNA goes into the DNA through this reverse transcription process. That was amply demonstrated by the study of retroviruses, particularly most famously through HIV studies that many of you know about. However, what could be well happening now, based on the work I just showed you, is that you can have genomic cDNAs produced in the same manner, except that it's not a viral gene. It's actually a normal invariant cellular gene. And it's not occurring in every cell. It's a mosaic phenomenon where it occurs in some cells rather than all of them. So what's this good for? Here's genomic mosaicism and somatic gene recombination in normal neurons. And here's the basic process. You start off with that genomic DNA. It forms that pre-mRNA before it gets spliced, before you remove the intronic sequences. And then it creates your RNA molecule. And then something happens. And this little blue Pac-Man is the reverse transcriptase that copies that RNA into a DNA molecule. And then this DNA molecule can go back into the genomic DNA to produce the genomic cDNA. So that's the basic process. So what's it good for? Well, maybe it allows you to produce activity dependent, and by activity, in neuroscience, we talk about activity in learning, memory, these types of phenomena that include gene transcription. This may, in fact, represent one form of activity-dependent recording of information, and it could do so with a sequence that's already spliced. So here's that spliced phenomenon that produces then the mature form of the mRNA. Why could that be useful? Well, it takes energy and time to splice out these introns. In some cases, you can have many, many, many introns and exons that need to be brought together. This process could produce a gene that was ready to go. It's already spliced, and have it produce its own RNA to produce a protein. Moreover, you may be able to change the way you express this gene. And what do I mean by that? Well, here we have that wild type, or the normal 2 copies, one from your mom, one from your dad, present in the genomic DNA. It's also associated with a way to control the expression of that gene, and it's referred to as a promoter. What's important is that's constant for a given gene. However, you may be able to change that so that you now have a different promoter, a different way of expressing that particular gene. Why might that be useful? Well, maybe it'll change the way that you as an individual, say, look at ice cream. So instead of liking vanilla ice cream, you now want Rocky Road. Or instead of being excited about, say, the color red, you're now excited about the color green. That there may be ways to stimulate, say, the expression in different ways of the same gene, but now it's controlled in a different way. Now, this playback reflects a form of memory. And in the nervous system, there's a whole bunch of different ideas that have been floated, such as something called a Hebbian synapse or Hebbian plasticity, neurons that fire together, wire together. Well, that might be a way you could take advantage of this. And you do so by changing the blueprint itself, by changing the very DNA sequence. Well, let's get back to Alzheimer's disease. And there's some oddities in the literature about Alzheimer's disease itself. where much of what you read, people say, oh yeah, you get Alzheimer's disease, that's it, you've got just Alzheimer's disease. But in fact, that's not true. That in the majority of cases, Alzheimer's disease is associated with other brain diseases. This is from one academic paper, where in addition to the Alzheimer's disease pathology, you also have Lewy body disease, which is associated with Parkinson's disease, other types of dementia. You have vascular dementias. And this is just one example. There are other brain disorders that are associated with Alzheimer's disease itself. So we were just talking about GWAS and those types of genes that have been identified for a given, say, Alzheimer's disease group. That completely ignores the simultaneous presence of these other completely different genes, different diseases, yet they're occurring in the same individual. Well, maybe if you had a somatic event, somatic gene recombination occurring and affecting other genes, not just APP, but affecting a bunch of other genes, that could explain the process to produce other types of pathology in the same brain. These are referred to as comorbidities. And so that is a formal possibility that we're currently investigating. Many of you have followed A-beta therapeutics, and the most famous of which are the antibodies that have been used to try to block these A-beta peptides. And if you look at a picture of the amino acids, here's the amino acid sequence of A-beta. And this is the same sequence that's been targeted by every single therapeutic. And what you can see are the names of those possible drugs. And the one that's been in the news most recently, this aducanumab by Biogen, is being looked at, and it interrogates this portion of the gene, of the peptide. But every one of these other ones has failed. And why could what I just told you about genomic cDNAs be relevant? Well, if you have a whole bunch of these little guys present, it's going to create other kinds of peptides. And because of the different single nucleotide variations that I also pointed out to you, it may be that these antibodies are not looking at the sequence or not able to interrogate these sequences because they're different. So these single amino acid sequences recognized by the antibodies may not be able to soak up all the other ones that are being produced by these small forms. There's some other repercussions, such as there are enzymes called secretases, as BASE is famous. None of these have worked either. Why haven't they worked? Well, maybe it's because you don't need cleavage by these secretases in order to have pathology produced by these small genomic cDNAs. And so here we have Alzheimer's disease through this new model of APP somatic gene recombination. And the way you can think about it is, rather than in the more controlled way that I showed you in neurons that might be involved in memory, it kind of goes wild. And what happens is that you form these genomic cDNAs, but then the process repeats itself again and again and again and again. Why is that relevant? Well, it turns out that reverse transcriptase is very, very well known to be a sloppy copier. And in fact, it is the reason for the escape of viruses, particularly HIV, from therapies, from its drug therapies, because it mutates. And in fact, we've heard a lot about mutation in the news lately for a number of other viruses, possibly even for COVID-19 related viruses. But the fact that these reverse transcriptases are sloppy copiers enables you to produce mutations. These mutations can accumulate, and they could form the seeds for these various plaques that are well known for Alzheimer's disease. And they could even form possible prion sequences. So what are prions? You've heard of these from mad cow disease. Well, there's a whole another line of evidence that suggests that proteins in Alzheimer's disease and other diseases as well may undergo a prion formation to make it transferable within the brain and perhaps between brains. Those prion sequences are not known, but here then is a mechanism that might produce them. Finally, there's a really interesting experiment, you could say, that medicine and nature has provided. and that's the HIV patient population. So HIV was a death sentence when it first came out in the 80s, early 80s. Those individuals uniformly would not survive, but with the advent of antiretroviral therapies, they live, and they live quite well, such as, say, Magic Johnson. And so they can live well beyond their 60s, and they are currently being treated by agents like the reverse transcriptase inhibitors. And so we can ask a very simple question. How many of these folks, the aged folks, have Alzheimer's disease? And this has been looked at in a variety of different ways. For example, the coming problem of HIV associated with Alzheimer's disease. And comorbidities of HIV were expected once again. I'm sorry, there's a phone going off. It'll stop at some point. And so HIV patients have been surveilled for a predicted avalanche of Alzheimer's disease cases over the past 14 years. So what would we expect to see? If we look at the numbers from the Alzheimer's Disease Association and from the CDC, these are some of the numbers, 1 in 10 over 65, 3% in the first strata, 65, 74, and it goes up from there. We get numbers that look like this. So say 100,000 or so folks are AD at risk today, that is, they're aged and have been on these reverse transcriptase inhibitors. And you would expect to see anywhere from, say, at the low end, 3% or some 3,000 folks, to say 17,000 in the higher age groups, or maybe even more if they're a lot older. But bottom line is there should be thousands of Alzheimer's disease cases in HIV positive populations. So what do you find? There's one case that's been reported, and that was in 2016, and this hasn't changed, of an Alzheimer's disease case in this HIV patient population that's been receiving reverse transcriptase inhibitors, arguing that this could be a very strong signal in looking for ways of interrupting Alzheimer's disease. There's actually an interesting corollary of that. Today, there are no approved disease-modifying therapies for AD, and these reverse transcriptase inhibitors are FDA approved now for HIV and actually for hepatitis B. And as a class, they've been used in humans since 1987. And so, you know, some 30 odd years of post-approval safety, that is, they're not something that you're gonna drop dead from taking it as a medicine. And there's nothing in current clinical trials today that will have this level of safety to help the 6 million Alzheimer's disease patients that are alive today. Now, legally in the United States, there are actually 2 compassionate use pathways for non-approved experimental agents, the so-called FDA Expanded Access Program and the Right to Try law that was signed into law in 2018. However, these aren't even relevant because reverse transcriptase inhibitors that are FDA approved can be prescribed off-label in the United States. And so, at least in theory, one could be prescribed reverse transcriptase inhibitors for the possible treatment of forms of Alzheimer's disease, albeit, of course, with many caveats, since we don't have all the details. There's been no prospective clinical trial work. We hope that someday, maybe not too long from now, such trials can be initiated. But it actually is an interesting sort of adjunct to the whole HIV pandemic, if you will, that from that arises a whole class of medicines that might actually be relevant to Alzheimer's and perhaps other neurodegenerative diseases. So with that, I've come to the end of my talk. Does DNA editing cause Alzheimer's disease? Well, maybe. And it's something that we can look at in greater detail in the coming years through genomic diversity via somatic gene recombination producing these pathogenic genes. So I thank you very much for spending some Saturday time with me, and I'd be happy to address any questions you may have.
Thank you very much, Dr. Chun. That was very informative. If we can get anybody to applaud here.
Yeah.
Jack, are you there? We can maybe, okay. So well, let's open the floor for questions. Since everybody's muted, you can't hear the applause.
Don't worry.
Okay. Let me see if I see any hands up on the list of people here. I'm not sure the hands up are being used properly. Is everybody, can everybody hear me? I guess if you have a question, use the raise hand feature to ask that question.
Now, Dorothy Parker has her hand up.
Okay, Dorothy.
Thank you, Dr. Chun. That was a very, very interesting talk. Do you have, I missed little bits of your talk because my husband was having Zoom troubles, but I think I got most of it. Do you have any ideas of why the Alzheimer's patients might have more of these reverse transcriptase related events and more of the cDNAs that are genomic?
Yes, that's a great question. And there's a number of hypotheticals, and probably the best way we have of thinking about it is actually cancer. And so what happens in a cancer? You have some type of a mutational event that occurs. And in fact, it's probably necessary to have multiple mutational events. There's the so-called two-hit hypothesis or carcinogenesis, where you not only need one mutation, you need another one or another insult. And that once you start that process, then you get this uncontrollable cell growth that forms a tumor, and then that forms, of course, the cancer. And so those are the notions in cancer. How would that work in the brain? So you've got these trillion cells, and each section or each portion of the brain actually has its own functional significance. You have parts of the brain dedicated to sight. You have parts of the brain dedicated to smell, parts dedicated to motor function, et cetera, et cetera. And it could be that there's certain regions within the brain that are more susceptible to these types of phenomena, and a mutation occurs, and it could be a whack on the head. It could be some genetic aspect that contributes, like we saw for ApoE4, that say increases the production of pathogenic genes like APP. Or it could be something else that we don't understand, such as something that someone has a seizure, someone has some type of life event, an IED, some explosive process. All of these are hand-wavy and hypothetical, but they have the potential to create that mutation. Once you create that mutation through this genomic cDNA process, which generates both copy numbers and mutations, then you start and accelerate over time the process of disease. And that's what we think may be going on, that you could actually be having an initial insult in a very small, or maybe just one neuronal population that then spreads. And we have there's some very interesting ideas about how this spreading could occur, but it also mimics the progression of disease. It turns out Alzheimer's disease, Parkinson's disease, many of these neurodegenerative diseases don't simply appear in your brain. They tend to start in certain regions, like the entorhinal cortex and things like that. And then they progress in a somewhat stereotyped way throughout the rest of the brain. That would fit in with a view that, yes, you start somewhere and then it spreads through the rest of the brain. So that's a concept we're pursuing. We'll see if it holds up.
Two other little questions along that line. One is that if a reverse transcriptase inhibitor helps, then the question immediately comes, some cancers have viral origins. Is there any chance that there might be a retrovirus or other virus that might be, and it might be triggered in some way by higher brain activity?
Yes, it's a wonderful question, and you're spot on, and with one modification, and that is that we do not believe it's an exogenous retrovirus infection that produces this, although formally that could be possible. But what we think is more likely is that it's endogenous retroviral types of activity. And what do we mean by that? Well, it turns out that our genomes today, as we sit here, about 50% of it arose from the actions of retroviruses and related viruses over the course of evolution. And there's hundreds of thousands of copies remnant copies, as they're called. And this fell into what has been termed junk DNA, that this stuff sits there and it's part of us. It can, in some cases, encode the very elements that would be relevant to this recombination process, and even more elements that might allow the spread of the disease. So we believe that there's, I think, a co-opting of some of these viral elements that our bodies has used over the course of evolution to produce us, to produce our brains, to use them. And then when you mess them up through various disease processes, that is still being used, but then it becomes pathological. But we are quite excited about the prospect that some of the very same types of elements that we find in the RNA viruses are present in some of these recombination processes.
Diane Withy? Diane?
All right. Yeah, I'd like to know about, my uncle married into a family that my grandfather said, don't marry into that family because they're all crazy. And his wife, her mother, and her brother all came down with Alzheimer's at a fairly young age. Does that have anything to do with, I mean, it runs in families. Does that have anything to do with what you're talking about, or how does that happen?
So part of the answer to that would depend on how young were these individuals when they developed Alzheimer's disease. If they were in their, say, 40s and 50s, then that falls into the familial forms of Alzheimer's disease that I mentioned, involving copies or mutations in APP or the presenilins. If it, excuse me, if it appeared much later, say in their 70s and 80s, that's more like a sporadic presentation. And then it could be just a coincidence, or it could be that you have some additional genetic propensity, such as those ApoE4 genes that we looked at, that then stimulate these genomic cDNAs. So both are possibilities, and part of it would depend on how early the onset was.
Yeah, I think the brother was fairly young and she was a little bit older, but I don't know about the mother. But yeah, it was devastating to their family.
Oh, yeah, it sounds horrible. Yeah.
Did Arlene have her hand up?
Yeah, yes.
Go ahead, Arlene.
I'm wondering if everybody should have their genes sequenced to see if some of these elements are visible, or should we all be examined at some point in our life to see if some of these diseases can be prevented?
Yeah, it's a question that has arisen about how you interpret these types of sequencing data. So in our case, you'd have to sequence your brain cells to see it. You couldn't do it by looking at blood cells. So in that case, no, it wouldn't matter. In the case of other diseases where you can detect changes in the blood cells, there's some merit to doing whole genome sequencing, as we call it. But for many, many, many cases, there's so many other variables that you can't interpret the sequence that you find. And so what it does is it, if anything, it scares people, or it makes them do things that they wouldn't have otherwise done because they think they might have a certain disease. And I think it's a big moral and ethical issue about how one deals with this type of sequencing information. In fact, at this very moment, there's huge discussions, even at the level of the National Institutes of Health, about how we use sequencing data from donors that give us, generously parts of their bodies, as say during autopsies. And the issue there is that an individual now must give specific consent to sequence information for us to be able to use the material, because sequencing is getting so powerful. You can identify people, individuals, based on their DNA sequence, and there are privacy laws that then come into play. So in terms of how that would, say, tell you about diseases that you would get, I'd say that it's not worth the effort to go about and do that, aside from curiosity. But if you do that, make sure you understand how someone like a company is going to use that information, because that could be used in the future for, you know, things that you might not want them to use it for. So that would be a bigger concern, not the disease identification.
Dorothy?
Yeah, I have another question.
Sure.
For sequencing of brain cells, that's done post-mortem. So
Yes.
you don't have to worry as much about a company. And that could be very valuable to brain research. So my question is there a place right now that a person could volunteer to provide samples? Because I think that could be very important.
Living samples?
No, I said post-mortem.
Oh, yeah. So
No, I'm
right,
not going to give, I mean, I'm using enough of my brain anyway, thank you.
Right. No, there's a number of programs that are available here in San Diego. UCSD has the body donation program, and it's just simply a matter of making contact with them, and there's some forms to fill out that relate to consent and some of these same issues that I just mentioned about open consent for sequencing. And it's very straightforward. And then what happens is then, you know, when one passes away, then the body is brought to UCSD and the brain removed and processed. And groups like our own would be notified, and then we'd be able to take advantage of that generosity to study. And so we, in fact, have put this into place at UCSD, which, you know, some of you will be amused that it took about 3 years and probably 30 bureaucrats spread throughout the University of California system, including system-wide and the president's office, and then another bunch that were somewhere in the hospital, somewhere on main campus. It was a nightmare of legal hassles, but finally we got through it. And now we have the ability to get that open consent. And I'm hopeful that this will be an important contribution from the community to understanding the brain.
Nancy?
Yes, thank you for that. I was just going to ask that question because we have filled out, my mother filled out the body donation program to UCSD. Yes. And I was wondering if your group would be able to get a part of it. My mother is currently living. She's 107 and a half.
Wow, it's amazing. You folks are amazing.
I hope I have her genes. I don't know. But so it's interesting to us. I mean, she started getting dementia. She's not terribly demented, but she started getting dementia about 100 years old. So she still knows who I am most of the time, and we can carry on a conversation fine. But I was just, that was my big question, was how would people like you get hold of it through the UCSD body donation? So that's wonderful.
Right. So the actual group that is there, the director's name is Scott Barton, Barton, and he has in his office are the forms for this specific use of open consent. And so if you just simply you sign it. In fact, it can be done post-mortem as well, as long as you are the executor of the estate or you have legal authority, then it can be done at that time as well. But the best thing that could be done for us would be to fill out the form ahead of time. That way, as soon as the unfortunate event occurs and someone passes, then we can immediately get an ambulance to bring the cadaver to the dissection suite and then isolate the brain. And it turns out that's super important because, as you know, as soon as someone passes, the body starts to deteriorate. So the sooner we can get access to it, the better.
I'd like to ask a question, then Dorothy. I see other people popping up, so yeah.
Sure.
I guess I didn't understand something. I'm sure I'm going to expose my ignorance. But basically, in cancer, as I understand it at least, some cell goes rogue, something happens, yes.
Yep.
It's not that that cell then goes and tells another cell, hey, I went rogue, why don't you go rogue too? What really happens is that cell begins to multiply and you see it.
Yep.
In the case of what you're describing with the brain, a cell goes rogue.
Yep.
But it's not, but Alzheimer's is not a cancer. So there must be a mechanism by which that particular cell now communicates to other cells to undergo the same process. Otherwise, you're really looking at individual cells that may or may not go off at given time. So how does that work?
Right. And that's a brilliant question. And it's what I'd alluded to about the viral type of mechanism. So neurons, by definition, are postmitotic. They cannot divide. What happens when a neuron goes awry? Well, it could be, say, hyperactive. That might give you a seizure. It could be dysfunctional. That might give you a psychiatric issue. It could die. That's neurodegenerative disease. So the endpoints, I think, are analogous, but the actual effect on a cell is quite different because it's postmitotic. But then how does that allow the transmission of that information, the pathological information from cell to cell? Well, neurons are actually the world's experts at transmitting stuff through their body and releasing it in two ways. They release it at sites known as synapses, where you have vesicles that then release contents that then stimulate other cells. That's point one. And point two is that there's another phenomenon called, they're called exosomes. And exosomes look like little viruses that can be released and are released by probably all cells at certain points in their life cycle. These exosomes can then transfer information to other cells, including those along a neuronal circuit. And there is evidence, some really exciting evidence for exosomal-like, there's lots of hand-wavy stuff, but exosomal-like processes that make it look like a virus. And so it could be, I don't know if Dorothy or someone asked earlier about the viral aspects. It could be that this transference is something that normally occurs through a mechanism that's viral-like, but then again, it goes awry in Alzheimer's disease. Now, there's one additional point that's quite fascinating, which is once you form these genomic cDNAs with these mutations on them, those mutations could give rise to weird proteins. And so I mentioned one group called the prions. The prions are actually known to be transmissible. This is the mechanism for Kuru, mad cow disease, Creutzfeldt-Jakob disease. And this is a mechanism where there's no DNA involved. It's the protein that then produces disease. So you now have a cell, or say a couple cells, that are spitting out these variant proteins that themselves then become prions. Maybe that's how it works, that you start to produce these pathogenic proteins that then self-replicate and create disease. So those are some of the variables we're investigating right now.
Thank you. Jan? Jan?
Yes, hi. So my question is, in the same way that peripheral cells turn out not to be, to reflect brain cells, is it possible that post-mortem samples do not reflect live brains? Is there any research comparing that?
It's always a challenge. And so we run controls and take extra steps to try to make sure that we're not looking at epiphenomena that are not part of that. And some of that comes from looking at vast numbers of different sequencing projects and finding the same things, and which is what we've done. We've gone and you can, and this is actually an interesting transition that's happened in biology, where you need the ability to interrogate terabytes of data that are floating around out there for sequencing discovery. And when you do so, then you start seeing these things that pop up in all these different sequencing databases. And it gives you the confidence that you're looking at something that's actually there in, say, a living cell line or in a living individual that came from a certain patient, in addition to the autopsy material.
Dorothy?
I just wanted to make a little announcement, and that is that Dr. Chun very kindly agreed to let his lecture be recorded, and that recording will be up on the SDIS website. And I'm sorry, I had something else right before this, so I could maybe put it on the chat, but I think instead, any of you who would like to know that information, if you would email me, or you can email the San Diego Independent Scholars. For that, the Gmail is just San Diego Independent Scholars, all one word, at gmail.com. Or you can email me at DorothyLParker@gmail.com. And I will send you the link to that information. Or you can just, if you go to the San Diego Independent Scholars website, you'll get the way to contact us, and you could ask to be put on our mailing list or be asked to get this information. And you can see some of our other recorded talks also.
Thanks, Dorothy. Yes.
No,
Oh, yeah. Go ahead.
am I on?
Yeah, you're on.
Okay, Dr. Chun, I looked at a report from the CDC, and it listed the mortality rates from dementia by different ethnic groups. The Asians had half the death rate as whites and blacks, and Indians, Native Americans, and Latinos were in between that. Do you have any idea why that's so?
It's a complex question, clearly. There's so many variables that go into that. But things like lifestyle and diet and could certainly contribute to some of that. In addition, there's well-supported documentation that educational achievement and say keeping active mentally, and in fact, every one of you here is proof positive of that, that it staves off dementia. And I'm not making light of that. I think it's, learning all your life and keeping active mentally is such an important phenomenon, as is physical activity that ties into it too. I think the fact that maybe societally we've divorced the two that's not good. It's all part and parcel of the same being. Your brain is integral to your motor function and your sensory function and everything else. So all of that, I think, feeds together to support activities, mental, physical, et cetera, that can promote better health in different groups. Now, I don't know from the CDC data about how that was stratified and what they controlled for, and other confounders, as they're called.
It's age adjusted, but that's all I read. But
right. So, for example, if you look at folks in the South, there's a lot of not so healthful activity that goes down there, and so that's one example, right?
Mm-hmm.
All right. Are there any other questions?
Yes. Can nobody see me?
Yeah, go ahead. You're on.
Okay, thank you. There are medications that have been given to people for many years, like Aricept, and it helps them in some regard, right? And a friend of mine's husband has been given that, and then for some reason, because it's taken internally, they've changed him to a patch so that it doesn't have to go through his digestive system. Does the function of those medications help you in any way, or is it completely unconnected to that?
Right. These are what we call, symptomatic or symptom treating medicines. They don't change the course of the disease. They can, in some individuals, make them feel better or function a little bit better. This particular one, Aricept, is a cholinesterase inhibitor, and it allows the neurotransmitter acetylcholine to function, at least in theory, better. But it doesn't, again, it doesn't change the inexorable course of the disease. That doesn't mean that it should not be used. It's an approved drug, and one wants to use every possible way to help a given patient. And so I'd say you still want to be able to have access to such agents, especially since they've been through some historically rigorous safety trials and clinical trials. But we still need things that actually work. So that's, I'd say it's part of the same, treatment goals.
And does age have a factor? What you're doing is that, would you, if a person is, say, 95, is that something you would still pursue, what you're working on?
Oh, well, you mean as a, say, a therapy, say a reverse transcriptase inhibitor, or do you mean the phenomenology of recombination? Is that still ongoing?
Yes.
Okay, yeah. So I think as long as you have an active functioning brain, this potentially could be ongoing. And I mean, it may have some kind of curious, also, I call them limitations. I think we were talking, Elvin had mentioned, or maybe Jack had mentioned about the hitting 120, that there might be a limit to lifespan. I know Tom was the counter argument, but let's say you think about your neuronal genome as say a recording medium, and how much recording can you do on a given tape or on a CD or something? Maybe at some point you reach a limit, and maybe that limit is related to the DNA state, how well it can be repaired, how well it can be accessed in order to get the information out of it. And so in that case, then maybe by the time you reach, say, 120, if any one of us would be so lucky to do that, then maybe you hit some sort of limits. I mean, that's just a speculation, but I think it's possible that there will be some biological limits that would be placed on that type of mechanism.
Thank you.
You're very welcome.
Yeah, Diana? Diana, are you there?
Yeah.
Could we ask people
this
to
is-
please use the raise hand where it's becoming an uncontrolled thing? David, I see you flailing, and I know you know how to raise your hand on the participant tab because you've used it before. Let's keep it an orderly discussion by please using the raise hand function in the participant tab.
I'm sorry, I can't. It doesn't work for me, so.
Okay, then David, why don't you go ahead and ask your question? And then
All right, I just wanted to ask Dr. Chun, are you far enough along in this idea and this hypothesis that you're ready to propose to the controllers of these things that some of the retrovirus controllers should be tried already? I mean, you and your colleagues are. Oh, I see. Have you initiated that process, or are you going to, or what?
Right. Absolutely, I think it should be tried. I think the issue is twofold. One is it costs a lot of money to run a clinical trial in Alzheimer's disease. Well, so we need to get funders to step up to actually try that. The problem there is that a number of these reverse transcriptase inhibitor drugs are off patent. And because of that, companies do not want to invest money in something that could be undercut by a generic drug. Now, there's two interesting, I think, side possibilities that might be helpful, and we're investigating it. One is outside of the US, where you have public health run medical systems, governmental medical systems. There, you want to keep people from becoming demented and having to be institutionalized. And so it might make good sense to try this in those countries because it could keep people functional longer. and that would have, you know, significant economic benefits. The other interesting possibility are folks that are suffering from familial disease or Down syndrome. And they are a more controlled population, a smaller population, but one where you would get a stronger readout. So you don't need as large, you know, they say a powered study that would be necessary for sporadic Alzheimer's disease. See, the problem with all of these types of sporadic cases is, a priori, you don't know who's going to get the disease. Someone that you stick in a clinical trial may never get the disease, but they're still part of that trial. Whereas if we looked at the families or at Down syndrome, everyone's going to get that disease. So that's a much more attractive group. It's also a much rarer and harder group to put into a trial, but you need smaller numbers. So I'm more hopeful that we might be able to move in that direction. I would love to get, and I've talked with many different clinical groups, and again, it comes back to money. They'll say, hey, yeah, we'll do it for you if you come up with, you know, 20 million bucks. And say, yeah, sure.
Okay, thank you.
You're welcome.
Diana?
Okay, this is just an observation. You might be familiar with this study on nuns that were in convents.
Oh, yeah. Oh, yeah.
Yeah, they followed them for many years and tested them regularly for cognitive decline. And they had agreed to give their brains to science when they died.
Yep.
Did this study, and they found that a lot of them had signs of Alzheimer's in their brain, but they never show cognitive decline in any of the tests because they always were active.
Yep, that's called a religious order study. And these, for those of you that aren't familiar with it, it's quite fascinating. These are cloistered nuns that enter the cloister when they're quite young, maybe in their 20s or thereabouts. And many of them are given tests at that point, simple writing tests. And the claim has been made that there may even be some indication of a predisposition to Alzheimer's disease based on their intellectual sophistication, the density of their thoughts, et cetera, as they write. But be that as it may, they're able to follow these folks longitudinally in the cloister and then have access to the brains. But they're very highly guarded and regulated. I've looked into trying to get access to some of that material, but I can't get access to it. So maybe in the future, you know, there may be some, you know, interest, but a lot of it, yeah, we were talking about earlier about the difference between academia and companies and different ways that, say, groups look at data. And yes, I think in general, you know, academia is a little more open, but I'll tell you, in this day and age where everyone's fighting for grants and everything, you know, people are pretty close to the chest when it comes to, you know, a strategically important resource like that. So, yeah.
Interesting. Okay, thanks.
Are there any other questions? Dorothy? Dorothy?
This is one more little announcement, and that is that, first of all, Dr. Chun, I don't know if you noticed, but every single one of the people who entered this meeting is still here. We still have the same number of participants. That is almost unheard of.
I'm flattered and honored. Honestly, I am.
Secondly, the San Diego Independent Scholars Science Group is going to be discussing your presentation and related papers a week from now. That's the group led by Arlene Gilbert, and if it's open to members and non-members, if you want to come to hear about this, we'll be talking more, going through some of these questions again more slowly. Maybe if you have some question or the science part of it you want to go over, that would be a chance to do it. And again, if you're not a member, if you're a member, you'll probably get a notice, but if you're not a SDIS member, then go to our website. It's just simply sdscholars.org, and go to our website. You'll find a way to contact us and ask to get the link to the Zoom meeting if you're at all interested. That will be next Friday at 1 o'clock.
Thank you, Dorothy. Okay, are there any other questions? Or this was pretty fascinating, and it's wonderful work. My God. So thank you so much again, Dr. Chun, for giving us this talk. It was wonderful, wonderful experience.
Well, thank you all. Please keep well and keep active like you're doing. You're, I think, an exemplary group, and may you have many, 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 many more years of lots of interaction. And please take care of yourselves.